Hypoxia, a state of reduced oxygen availability, is a key factor influencing cellular and systemic functions in both physiological and pathological conditions. Epigenetics, encompassing DNA methylation, histone modifications, and non-coding RNAs, mediates the cellular response to hypoxia by modulating gene expression without altering the DNA sequence. This technical note provides an overview of the mechanisms and significance of epigenetic regulation in hypoxia, drawing from findings in recent studies. Hypoxia-inducible factors, HIFs, are central to hypoxia signaling, and their activity is regulated in part by DNA methylation. Hypoxia alters the activity of DNA methyltransferases, DNMTs, leading to changes in the methylation status of CpG islands within promoters of genes critical for adaptation. For instance, hypoxia-induced hypermethylation of tumor suppressor genes can contribute to cancer progression, while hypomethylation of angiogenesis-related genes enhances vascular remodeling. Hypoxia has been shown to upregulate DNMT1 expression in several cancer types, including breast and lung cancers. This upregulation contributes to hypermethylation of tumor suppressor gene promoters, such as CDKN2A, P16, facilitating unchecked cellular proliferation. Hypoxia enhances DNMT3A and DNMT3B expression in some cancer cell lines, promoting de novo methylation of CpG islands associated with key regulatory genes. For example, hypoxia-induced DNMT3B activity in hepatocellular carcinoma is linked to the silencing of the E-cadherin gene, promoting epithelial to mesenchymal transition, EMT, and metastasis. Similarly, DNMT3A has been implicated in the methylation of metabolic genes under hypoxia, reshaping cellular metabolism. Studies suggest that hypoxia-modulated DNA methylation patterns are cell-type specific and temporally dynamic, emphasizing their role in diverse physiological and pathological contexts. Histone acetylation and methylation are pivotal in regulating chromatin structure and gene accessibility under hypoxia. Enzymes like histone acetyltransferases, HATs, and histone deacetylases, HDACs, along with histone methyltransferases, mediate these changes. Hypoxia often leads to a global reduction in histone acetylation, but site-specific modifications enhance the expression of hypoxia-responsive genes such as VEGF and GLUT1. Hypoxia upregulates HDAC1 and HDAC2 expression, particularly in cancer cells, where they interact with transcription factors such as HIF1-alpha to repress pro-apoptotic genes like BAX and PUMA. HDAC1 also plays a role in compacting chromatin structure, thereby silencing genes that counteract tumor growth. HDAC3 is activated under hypoxia and contributes to the repression of differentiation-associated genes, maintaining a dedifferentiated state in cancer stem-like cells. This is particularly evident in glioblastomas, where HDAC3 cooperates with HIF1-alpha to regulate stemness-related pathways. HDAC6 is upregulated in hypoxic environments and is associated with increased angiogenesis and tumor invasiveness. HDAC6 deacetylates alpha-tubulin, which enhances cellular motility, facilitating metastatic spread under hypoxic conditions. Histone demethylases are enzymes that remove methyl groups from histones, dynamically regulating chromatin accessibility and gene expression. Specifically, Jumanji C histone demethylases are a family of iron 2 and alpha-ketoglutarate-dependent enzymes, making them directly sensitive to oxygen availability. For example, hypoxia reduces the activity of JMJD1A due to limited oxygen availability, leading to an accumulation of methylated histone, H3K9Me2. This represses the expression of genes involved in differentiation and energy metabolism. JMJD2 enzymes are inhibited under hypoxia, leading to increased H3K9Me3 levels and reduced accessibility at certain genomic regions. Hypoxia-driven JMJD2A downregulation has been linked to altered DNA repair mechanisms in tumor cells. Overall, the interplay between HIFs and histone-modifying enzymes further influences chromatin remodeling, establishing a direct link between the hypoxia signaling pathway and epigenetic control. Non-coding RNAs, particularly microRNAs and long non-coding RNAs, act as crucial regulators of the hypoxia response. Hypoxia induces specific miRNAs, such as MIR210, which target genes involved in mitochondrial metabolism, cell cycle regulation, and apoptosis. Similarly, hypoxia-regulated LNCRNAs influence chromatin organization and transcriptional activity. For example, hot air has been implicated in promoting metastasis in hypoxic tumor environments by interacting with chromatin-modifying complexes. Key epigenetic enzymes, such as 1011 translocation, TET, proteins that demethylate DNA and Jumanji C domain containing histone demethylases, are sensitive to oxygen availability. These enzymes require oxygen as a cofactor, and hypoxia can directly inhibit their function. For example, the reduced activity of TET enzymes under hypoxia leads to an accumulation of 5-methylcytosine, altering gene expression patterns associated with cellular adaptation and survival. Understanding the epigenetic landscape in hypoxia has paved the way for developing drugs targeting these modifications. HDAC inhibitors and DNMT inhibitors are under investigation for their ability to reverse hypoxia-driven epigenetic changes in diseases such as cancer and ischemic disorders. Additionally, strategies to modulate hypoxia-responsive miRNAs offer a promising avenue for therapy. Despite significant advances, much remains to be explored regarding the temporal dynamics and reversibility of hypoxia-induced epigenetic modifications. Emerging technologies such as single-cell epigenomics and CRISPR-based epigenetic editing hold potential to unravel the complexity of hypoxia's epigenetic regulation. Further, integrating epigenetic profiling with clinical data could lead to precision medicine approaches tailored to hypoxia-associated pathologies. This body of evidence underscores the critical role of epigenetics in mediating cellular responses to hypoxia and its implications for health and disease. Future research will likely expand our understanding, leading to novel therapeutic interventions. Learn more about Embryant solutions for hypoxic cell culture.